Every living cell, whether in humans, plants, or bacteria, is built from tiny chemical molecules. But among all the biomolecules, one group does most of the real work of life. They build tissues, they run reactions, they carry oxygen, they send signals, they even defend us from diseases. These molecules are made from simple building blocks called as amino acids. In this video, we'll clearly understand how amino acids form peptides, peptides form proteins, and proteins make life possible. Let's start from the very basics. Life is all about chemistry, right? So all living organisms are made of four major classes of biomolecules: carbohydrates, lipids, proteins, and nucleic acids. Among these four, proteins are the most diverse and functional molecules. They are built from small repeating units, just like beads forming a chain. One important rule or guide to remember is that the structure determines the function. The shape of a molecule is what decides what it can do. So to understand proteins, we should first understand their smallest units, which are amino acids. Amino acids are small organic molecules that act as building blocks of proteins. Each amino acid has two important chemical groups, an amino group and a carboxyl group, which is attached to a central carbon called the alpha carbon. They also have a side chain or R group, which makes each of the amino acid different. In nature, there are 20 standard amino acids, just 20. But by arranging these 20 in different sequences, cells can create thousands of different proteins, each having its unique functions. So you can think of amino acids like as letters of an alphabet. 20 letters, but makes infinite amount of words. Not all amino acids behave the same way. They are classified based on the properties of their side chains. So some are non-polar or hydrophobic, meaning they avoid water. Some are polar but uncharged, able to form hydrogen bonds. Some are charged, either acidic or basic, and few are aromatic or have special cases with unique structures. These differences are very important because these chemical properties is what determines how the proteins fold and what is its function later on. So amino acids also have some of the most interesting chemical properties. They are amphoteric in nature, which means they can act as both acids and bases. Depending on the pH, they can gain or they can lose protons. At low pH, they carry a positive charge. At high pH, they carry a negative charge. And at a specific pH called as the isoelectric point, the net charge becomes zero, and this form is called as the zwitterion. So because of this property of zwitterion, amino acids exist in this zwitterion form. Because of this property, amino acid also act as biological buffers, helping to maintain a stable pH inside the cells. They also can form hydrogen bonds and ionic interactions, which later help to stabilize protein structures. So chemically, amino acids are very dynamic molecules. Another important classification is nutritional. So amino acids are essential. Some amino acids are essential amino acids, which means our body cannot synthesize them. So we should obtain them from food. Okay. Then others are non-essential amino acids, which is the body can make it on its own. So both types are very important for growth, repair, and metabolism. A deficiency in essential amino acids can affect protein synthesis and the overall health. So that is why protein-rich diets are so important. So now the big question is that why are amino acids so important for life? Because amino acids, they are not just the building blocks of proteins. They are involved in almost every aspect of biological process. So first they build together, they join together and they form proteins, which perform most of the cellular functions. Secondly, the amino acids act as precursors for numerous hormones, right? They act as precursors for hormones. And for example, many signaling molecules in your body, they originate from amino acids. So they also form neurotransmitters, which help the brain cells to communicate and control thoughts, mood, and memory. In addition, amino acids contribute to metabolites, energy production, and biosynthesis pathway. So from muscles to hormones to brain signals, amino acids are everywhere. That's why amino acids are one of the most fundamental molecules of life. Now let's see how the amino acids join together. Fine. So when two amino acids react together, the amino group of one joins with the carboxyl group of another. During this process, one molecule of water is released. This is called a condensation reaction. The bond that is formed is called a peptide bond, also called a amide bond. So by repeating this process again and again, amino acids form long chain. This is how peptides and proteins are created. Now that we have understood how peptide bonds form, let us see how amino acids assemble into larger chains. So when just two amino acids join together, they form dipeptides. Okay. If three of them join together, they are tripeptide. If a few amino acids link together, usually less than 10, we call them as oligopeptides, meaning short chains. But when many amino acids connect in long sequences, sometimes hundreds or thousands, we call them as polypeptides. These long chains are very important because they can fold into complex shapes. Only after folding do they become functional proteins. So remember this, that the simple idea is that small chains are peptides and long chains are polypeptides. And the folded polypeptides become proteins. So what exactly are proteins, right? Proteins are basically long polypeptide chains that have folded into three-dimensional shapes. Okay. So they are built using 20 standard amino acids. They are arranged in different sequences like letters forming different words. But what makes proteins very special is not just the length, it's the shape. Each protein folds into a unique 3D shape, 3D structure. And this 3D structure determines what it can do. And what do they do, right? Almost everything. Okay, proteins can act as enzymes. They can speed up reactions. They can be antibodies fighting infections, transporters like hemoglobin to carry oxygen, structural proteins like collagen to give our body strength. That's why proteins are often called as the tiny machines of life. To understand protein better, we should understand their levels of structure. Proteins are organized into four levels, okay, of structure. Four structure levels are there in proteins. First is the primary structure. This is simply the sequence of amino acids like beads on a string. Next, you have the secondary structure. Here, the chain forms local patterns like alpha helixes and beta sheets, and they stabilize through hydrogen bonds. So then we have the tertiary structure. This is the complete 3D folding of a single polypeptide chain. So there are interactions like hydrogen bonds, ionic bonds, hydrophobic interactions, and disulfide bonds help maintain this shape. Finally, some proteins have quaternary structure where the multiple polypeptide chains they assemble together and they form one functional unit. So example is hemoglobin. Okay. So in short, you can say that the sequence gives you the folding and the folding gives the shape and the 3D shape gives you the function. So let's quickly visualize everything together. We start with simple linear sequence of amino acids. That's the primary structure. Small regions, then they form helixes and sheets. This is the secondary structure. These elements they fold to further into a compact 3D shape, a 3D structure, forming the tertiary structure, and sometimes several folded chains combine and they form multi-subunit complex, which is the quaternary structure. Right? So this step-by-step folding is extremely important because if a protein folds incorrectly, it will lose its function or it can even cause disease. So the key idea is that structure controls function. And that's why protein folding is one of the most important concepts in biochemistry. Now that we know proteins should fold into specific shapes or structures, an important question comes up. what keeps that shape stable? Fine. Proteins don't stay folded by chance. They are held together by several small chemical forces which work together. First, there is hydrogen bonds. So these are weak attractions between slightly positive hydrogen atoms and electronegative atoms like oxygen or nitrogen. Even though each bond is weak, thousands of them are held together. They held together strongly. They stabilize the alpha helices and beta sheets. Second is the ionic interactions. They are also called as salt bridges. Here the positively charged side chains, they attract negatively charged ones like magnets. So they help lock the parts of the protein together. Third is the hydrophobic effect. So non-polar amino acids, they avoid water and they pack tightly inside the protein. They form a hydrophobic core. This is actually the main driving force for protein folding. Finally, there are the disulfide bridges. So these are strong covalent bonds which form between two cysteine residues. They act like molecular staples, so they add extra strength and stability. So remember that many small forces together create one stable protein structure. Now let's understand protein folding. When a protein is first synthesized, it start has a long linear chain which is called as an unfolded polypeptide. But this chain is not functional yet. It should fold into a precise 3D shape or 3D structure. This is called as native conformation. Only after the folding does the protein become active and capable of performing its job. Inside the cell, special helper proteins called chaperones, which are there, they assist folding. Fine. They prevent the misfolding and aggregation. They ensure that the proteins reaches the correct structure. However, folding is very delicate, right? So if you apply heat, extreme pH, or harsh chemicals, these stabilizing forces break. The protein unfolds or denatures. And when the shape is lost, the function is lost. So the key message here is very simple, is that structure determines function. Loss of structure, loss of function. Now let's see why proteins are so important. Think of proteins as tiny molecular machines that are there inside every cell. Each protein has a specific structure that allows it to perform a specific task. Some proteins are enzymes. They speed up chemical reactions that keep the life running. Some are structural proteins. They give strength and shape to cells and tissues. Others help in transporting, example like oxygen ions or nutrients across the membranes. And many are involved in signaling. They receive messages and coordinate cellular responses. So proteins aren't just molecules. They are workers, builders, messengers, and defenders of the cell. Without proteins, life simply cannot exist. To conclude, let's appreciate the functional diversity of proteins a bit. So from just the 20 amino acids, cells can create thousands of different proteins, each having a unique role. So as I told before, as I mentioned in the previous, some can become enzymes, catalyzing almost every biochemical reaction. Some provide structural support, like collagen in skin or keratin in hair. Some handle transport and signaling, like hemoglobin carrying oxygen or receptors transmitting signals. Some protect our defense, act as immune defense molecules, like antibodies, which recognize pathogens. So, from one simple idea, amino acids joining into proteins, we get incredible biological complexity, which is the power of proteins. They truly are the functional foundation of life. Among all proteins, enzymes are one of the most important and fascinating ones. Enzymes act as biological catalysts, which means they speed up chemical reactions inside the cell without getting consumed themselves. Now you might wonder, why do reactions need help, right? Every chemical reaction requires a small amount of starting energy to begin. This initial energy barrier is what is called as activation energy. So you can think of it like pushing a ball over a hill. The ball needs an initial push to reach the top before it can roll down. Enzymes help to lower this energy barrier, making it easier for the reaction to occur. Because of this, reactions that would normally take hours or even days can happen in milliseconds inside living cells. Enzymes are so specific, highly specific. Each enzyme usually works with only one particular substrate, which it is like a lock and key. And since they are reusable and regulated, they allow the cell to control metabolism very efficiently. So without enzymes, life would become very slow to exist. Now let's look at some of the real life examples that show how important biology is, how important biological rule, which is structure determines function. The shape of protein directly decides what job it can do, what function it can do. First, we have collagen. Collagen forms a long rope-like fibers. They are extremely strong. Because of the stretched fibrous structure, it provides mechanical strength to skin, tendons, and bones. So its structure is designed for support. Next, you have hemoglobin, a globular protein. It is found in red blood cells. Its compact folded structure creates pockets which will help bind oxygen molecules. This allows hemoglobin to efficiently transport oxygen throughout the body. Then we have antibodies. So their Y-shaped structure is not random. It allows two arms to specifically recognize and bind to foreign particles like bacteria or viruses. So this makes them perfect defense molecules for immunity. Finally, motor proteins such as myosin act almost like tiny walking machines. They move along filaments inside the cell and generate force, allowing muscle contraction and cellular movements. So from fibers to globules to moving motors, each protein structure is beautifully matched with its function. That's true. That's the true power of protein design. Now that we have seen how protein structure defines function, let's see how scientists actually isolate and study these proteins in the lab. After understanding what proteins do, an important question arises. How do scientists actually study these proteins in the laboratory? Inside a cell, there are thousands of different proteins that are mixed together. So we should first isolate one protein at a time to study its structure and function. The process starts with a crude extract. So you break open the cells and you release all their contents, proteins, DNA, lipids, everything is mixed together. So next we should clarify the mixture. So you use centrifugation. Heavy debris and cell fragments are removed. It gives you soluble proteins in the solution. Then comes the separation techniques. So techniques like chromatography helps to separate proteins based on properties like size, charge or binding ability. So this step gradually it enriches the protein we want. After that we analyze. So the analyze the sample. So you use methods like electrophoresis or spectroscopy and you check the purity and activity. Finally, after we obtain a pure protein, which is ready for detailed study, structural analysis, or even therapeutic use. So protein research is not just theory, it involves careful experimental steps which allows us to isolate and understand these incredible biological machines. After purifying our protein, we must use specialized analytical techniques and we should study its properties and structure. So now let's look at the overall workflow of protein purification, how it occurs, the step by step. Okay, the overall protein purification workflow, step by step. So everything starts with cells because proteins exist inside cells, so they have to be opened up. They have to be broken down, broken up. Okay, opened. So the first step is cell lysis, so where we break open the cells and release all the contents. So there will be proteins, lipids and other molecules. So this is a crude mixture. This is called as a crude extract. Next comes fractionation. So here you use techniques like centrifugation and you separate the heavier debris from soluble proteins. This clarifies the sample and removes unwanted material. After that, you perform chromatographic separation. Here proteins are separated based on properties like size, charge or affinity. This step gradually enriches the protein that we want while you remove the others. Then we check the activity and purity using gels or assays. We then confirm whether the protein is functional and whether only one band, which means one protein is present. Finally, we get the pure protein, which can be used for structural studies, experiments, or even medical applications. So protein purification is like literally filtering step by step from a complex mixture to one single clean functional molecule. And only after purification can we truly study how that protein works. Once we have isolated the proteins from cells, the next step is to study and analyze them. Because proteins are invisible to the naked eye, scientists should use special laboratory techniques to separate, measure, and understand them. The first common method used is electrophoresis. In this technique, proteins are placed in a gel and an electric field is applied. Depending on their size and charge, they move at different speeds. So smaller proteins will travel faster, larger ones move slower. This helps us to separate proteins and check their purity. Next is chromatography. Here proteins pass through a column which is filled with a special material. Based on the properties like size, charge, or binding ability, each protein will move differently and separate into fractions. This method is extremely powerful for purification. Then you use spectroscopy, such as spectrophotometry. This technique measures how much light a protein solution absorbs. Right? So it helps us to determine protein concentration quickly and accurately. Finally, for detailed structural information, you use advanced methods like X-ray crystallography or mass spectrophotometry. So these techniques allow us to see protein structures at atomic resolution or determine their molecular weight and composition. So together these tools will help us to answer three main questions. One is that how much protein is there? Second is how pure is it? And third is what is it what is its structure? Fine. So that's how we truly understand proteins scientifically. Fine. So far we have understood the chemistry, structure of amino acids, proteins, peptides. But an important question remains is that why does all this really matter in everyday life? Why are we studying all this, right? So the answer is very simple. Proteins directly impact our health, medicine, technology. In nutrition, the proteins we eat simply they supply your essential amino acids. They help build muscles, repair tissues, and maintain overall body. In medicine, many life-saving drugs are actually proteins. For example, insulin is used to treat diabetes, and vaccines or antibodies help protect us from infections. In diagnostics, protein-based tests allow doctors to detect diseases early through blood tests and biomarkers. And in biotechnology and industry, engineered proteins are used to design drugs, produce enzymes, and even create new therapies. So proteins are not just molecules that you study in textbooks. They are actively improving and saving lives every day. This is where biochemistry connects directly to the real world. So now let's summarize everything that we have learned. We have started with amino acids, the smallest building blocks. When amino acids join together, they form peptides. And when peptides fold into complex three-dimensional structures, they become proteins. And these proteins perform nearly every important function inside the cell. One key principle we emphasized throughout is that structure determines function. The sequence of amino acids decides the structure, and the structure decides the job the protein will perform. From enzymes and transporters to antibodies and hormones, proteins drive life processes. That's why proteins are central not just to biology, but also to medicine, biotechnology, and research. So understanding protein means understanding life itself. To conclude, let's step back a bit and look at the bigger picture, right? Life begins with simple molecules, actually. Amino acids, they combine, they form peptides. Peptides fold and they form proteins. And proteins create complex machinery that makes life possible. Every heartbeat, every breath, every thought depends on proteins working silently inside our cells. So something as small as an amino acid ultimately leads to something as complex as life. And that's the beauty of biochemistry, tiny molecular interactions creating the miracle of life in living systems, right? Thank you for learning with me. I hope this session helped you to see how amino acids, peptides, and proteins truly form the foundation of life. Thank you, everybody.